So I'm actually giving the third talk as well here this morning. This is a talk, a totally different talk, incidentally, about some other compounds that we've been working with that are fairly interesting as well, and I hope you're going to enjoy this. So this talk is titled Liamycins from Oribasidium pullulans with antibacterial activity specific for species of Streptococcus. So, Oribasidium pullulans is actually a soil-dwelling yeast. It's been used for a very long time. Commercially, it's used for the production of a polysaccharide called pullulans, and this pullulans is used to make these breath freshener films. I don't know if you've seen these films that have sort of mint. You put them on your tongue, and they dissolve up on your tongue. They're these things. They're actually made by Johnson & Johnson. So, Oribasidium pullulans has been known for a long time because of that. And pullulan itself has been made commercially since the mid-70s because of that. We weren't particularly interested in that aspect of this microorganism, but we had some other interests. A guy at the lab, Timothy Leathers, had some interest in other compounds that were being made. And when we were growing this in fermentation, what we found was rather interesting. This is the Oribasidium pullulans yeast growing in fermentation. And you can see the accumulation here of a fluorescent yellow oil in the bottom of the fermentation vessels. And in fact, it's probably made, it's probably made from a biofilm that it starts to form around the fermentation flasks. And also, if we look under a microscope at the cells, you can actually see it fluorescing, and it's probably in the vacuoles of this yeast species. So we found this compound, and we were sort of interested in this compound, and we analyzed this compound, and we published the structure a few years ago, back in 2013. This is a multi-TOF spectrum. The oil itself actually consists of 4 compounds that we call A1 and A2, B1 and B2. And what those compounds are, they're a mannitol head group, and they have attached to them these fatty acid chains at one end of the polyol. So mannitol is modified at just one end with this 3,5-dihydroxy fatty acid chain. And then we found there are actually trimers of 3,5-dihydroxy fatty acid, or there can be tetramers of this 3,5-dihydroxy fatty acid. And in fact, the difference between the A1 and the A series and the B series is that one fatty acid chain. And the difference between A1 and A2 is actually an acetate group. The 42 mass units here is acetate group. So there is an acetate at this position here and here. And we determined all of that by mass spec. This is by NMR. And this is all published. So what wasn't published until very recently was this finding of this anti-streptococcal activity. And I just want to go through that. This is rather an exciting finding, we think. So we took the oils, the liamycin oils, the crude oils, and we separated by reverse phase HPLC. So you're just looking at an HPLC profile up there. And we take fractions from the HPLC, and we assay them by multi-TOF mass spectrometry. So what you're looking at here is you're looking at individual liamycins. So the A1, the A2, B1, and B2. This is the A1, the A2, B1, B2. And here, actually, you can see a little bit of pentamers, which is sort of interesting. And up here, something that I really want to talk about a little bit here is some dimers. All right, dimers meaning 2 fatty acid chains attached to the mannitol. And down below, what you're seeing down there is just an assay on a agar plate. And we have the agar plate growing on this plate. We have this Streptococcus agalactiae. Okay, and then so we're looking at, this is the control. This one here, you can see the zone of inhibition. This is just the crude oil prep showing that it kills the Streptococcus. And then as we start to look at these fractions 1 through 4 here, and then 5, 6, 7, and 8 here, and then 9, 10, 11, 12, and then 13 and 14 on these agar plates. And you can see that the early fractions have very little activity whatsoever, and then there's not so much activity there. And then we're starting to get activity and then more activity, and then here we're getting quite a reasonable zone of inhibition of cell death. And another way of looking at that is that we can do a broth dilution and we can just look at the minimal inhibitory concentration for the same streptococcal strain. And you can see the dimer here, actually, that says 128 micrograms per mil, or micromolar since it's 1,000 molecular weight. So the dimer has very little activity. It seems that the dimer has very little activity. And then the trimers are around about the same sort of activity. And then we're looking at about 14 micromolar for this tetramer without the acetate group on there. So the most active compound is this mannitol liamosine B. And it was most active against Streptococcus agalactiae. This was just the strain that we were using to identify the antibacterial activity. So we went back to a number of different bacterial strains, and we found very good activity against a bank of 9 different Streptococcus. So we're looking at agalactiae. This is micrograms per mil for the minimal inhibitory concentration. 20 micrograms per mil for the against agalactiae. We're getting fairly good inhibition against mitis, good inhibition against Streptococcus mutans. That's the one that causes tooth decay. One of which is very important to the USDA is Streptococcus suis, which is a very important pathogen of pigs. It's also an emerging pathogen in human beings as well, as it turns out. And here actually we have a very good activity against this bacterium as well. What is also interesting is that one of the strains here, the Sabronicus strain, actually has very little activity whatsoever. And if we look at Enterococcus, moderate sort of activity. Lactobacillus, Staphylococcus, E. coli, Pseudomonas, no activity at all. So we had what seems to be a selective antibacterial activity against certain Streptococcus strains. Okay, so we actually then used our MALDI-TOF there as a MALDI-TOF-based screen. I don't want to go into this in too much detail, but we actually screened 54 different strains of Aurea basidium. There was a postdoc who went out to Thailand to isolate these things, and we found that 21 of them produce liamosines, and you can see that some of them here, we're just looking at a profile of A through J, whatever that is in numbers, of some MALDI-TOF profiles from oils from these different strains. But what I just want to show you is this here is A1 and this is A2. So some of the strains were producing quite a lot of the A-type liamosines, and then other strains were producing very little of that, but then would produce a lot of the B-type liamosines, the more active form. And if we look at actual overall production, you can see the yield is really quite good. We're talking about 5-6 grams per liter in some of these strains, and typically 2 grams per liter. So this is excellent, much better than sort of micrograms of material. And you can see these oils here, they're just grown in different culturing conditions. This strain also makes a siderophore as well. So we saw quantitative differences between the different components that were present, but essentially not too many structural differences. Okay. So then we did this experiment. And so this experiment, we grew one of the strains on a minimal medium, but we had a single carbon source in there for growth. So we used all of these different sugars. And then what we did is we looked at the polyol head groups from the acid hydrolyzed liamosine. So we just take the liamosine, do an acid hydrolysis, and we found no matter what we grow them on, we find mannitol as the head group. Okay, I already told you that, so why am I telling you this? This is boring. What was not boring is that we then grew them on a set of different polyols. So when we grow them on different polyols, we find a selective incorporation. If we grow them on D-mannitol, we get mannitol type. If we grow them on arabinitol, we get 98% arabinitol as the head group, and we do not get very much of the mannitol type, only 2%. We tried various other sugars and we found different amounts of incorporation. So we see if we use sorbitol, we get a sorbitol type compound, but the default position seems to be to have a mannitol head group. I want to just show you this one and this one at the bottom here, 3-etol 4-carbon polyol. We get 78% 3-etol type, and if we use glycerol, we get 75% glycerol type. Okay, another way of looking at this is if we use the MALDI-TOF. So at the top here, we just grow the strain on mannitol, and we get the 4 compounds I showed you before. So these are the mannitol type liamosines, A1, A2, B1, and B2. If we grow them on arabinitol, we get now arabinitol type compounds and no mannitol compounds whatsoever. So these are 30 mass units less. If we grow them on the 3-etol, we get 3-etol type compounds. If we take this compound and we run them on a chiral GC column, we can see now that these are the controls. So this is D-arabinitol and L-arabinitol. And what this is showing you is that the arabinitol, which is incorporated into these compounds, is actually just the D stereoisomer. And another way of looking at that is like this. If we grow if we grow the Aureobasidium on D3-etol, we get mannitol type. If we grow it on L3-etol, we now get 3-etol type. Okay, so 3-etol, the L is being selectively incorporated. If we look at the arabinitols, the L-arabinitol gives us mannitol type, the default position. And if we grow them on D-arabinitol, we now get new compounds that are being formed with a arabinitol head group on there. All right, so then we wanted to think about the specificity of these compounds and whether we have any difference in the selectivity of these different head groups and whether that might affect the biological activity. And the answer is that they're all active against Streptococcus. So we looked at Streptococcus agalactiae, and what we found is that the D-mannitol type of liamosine is more active. So if we have a mannitol head group here, and we have 3 or 4 fatty acid chains, they're very selectively active down into the low micromolar range. To give you some sort of an idea, penicillin is probably active in about 0.1 micromolar range, so it's about 10 times less active than penicillin is against this particular strain, but highly selective. Okay, we found they were actually very highly selective, and we do not see activity against Staphylococcus, for example. The other thing which is very curious, and we think perhaps telling, is that if we have the dimer, which only has 2 acyl chains on there, we have no activity whatsoever. Right? The dimer loses activity completely. So we now have a arabinitol type, and we have a 3-etol type, and we have several others that we're working on, of course. I just want to show you a couple of other compounds which are kind of interesting. They were isolated a number of years ago in 2 different labs. In 1996, 2 compounds isolated. This essentially is just the fatty acid component of these compounds, and it's called exophilin A. It has antibacterial activity against a bank of bacteria, but the activity against the single Streptococcus that was tested was actually very low indeed. So there was activity against Enterococcus, and there was activity against Staph, and we do not find that. So there is quite different activity without this polyol head group in position. And then these compounds over here, these halimesins, they have a mannose at the other end, mannose as opposed to mannitol. And again, the activity, the antibacterial activity is quite different. The bank of activity is quite different. One final piece of data. This is a live dead assay. It actually is one of these fluorescent assays. The cells are, it's a kit that's sold. Live cells are green, and dead cells are red, and you can look at live dead components. So we can do this within 1 hour after the treatment, and it allows us to very quickly determine the cure rate. So again, we use Streptococcus agalactiae for this strain. These are the 2 controls. This is just saline and DMSO. Excuse me. And you can see here, 50% cure rate at 20 micrograms per mil. And by 80%, around about 80%, 80 micrograms per mil, we have a 100% cure rate. And we can also plot that in a different way just by counting colonies on a plate there. So that's just a separate piece of information on the same there. So very rapid kill rate within about an hour, and very low activity, concentration disruption of membranes probably. Okay, just to finish this talk, my thanks to Ken Bischoff again, the antimicrobial guy at the lab, Tim Leathers, who works with the Aureobasidium, Penn, who went out to Thailand, I'm losing my voice here, and Joseph Rich, who runs our research unit. And this guy here is the origin of the name liamosines. Actually, this is my son. my eldest son. I just thought I'd thank him for this. Even though that caused a problem with the talk, that the talk didn't want to load up properly when I put his photograph in. All right, thank you, and I'll ask for questions.
A very interesting talk, and I'm interested in this mammalian as an antibacterial. Have you considered trying this on strains of bacteria, for example, Streptococcus, that are antibiotic resistant? Mm-hmm. And would that overcome this antibiotic resistance and kill it?
Yeah. Okay, this is an excellent question. Okay, what we don't have is a mode of action. And clearly, this is interesting, but are we really inventing a new type of soap or a new sort of detergent? So this really is the question. And the way to approach that is maybe a couple of different ways, meaning I'm not answering his question, but I'm going to kind of hedge around it towards it. So the first thing is that we found one of the Streptococcus was not hit by the liamocins at all. It seemed to be very resistant. Right, so one way of looking at that is, well, it's selective for the other types. There's something in this strain which is extra that's blocking this, or there's something in this strain that's missing. So one way of doing that, again, excuse me. What we're trying to do there is to take a genetic approach and perhaps either to make knockouts that are that become resistant. The alternative approach is to take that strain, clone whatever it is that's in that strain that's causing the resistance into the galactase strain. Have we done those experiments? We're starting those experiments. Right? So we're looking at a knockout library of Streptococcus, and we're going to try and select for bacterial resistance and then work it out that way.
Do you have any idea what the fluorophore is?
Yes. Okay.
And even if it is fluorescence, could it be phosphorescence?
It could be riboflavin. It's possible it's riboflavin. So in the we actually were able to isolate that compound in the HPLC runs, and it's fairly water soluble. It comes off a lot before the liamocins does, and in some of the strains it doesn't seem to be present. In the strain that we did most of the work on, unfortunately, it was present. And in some ways that was great. It meant we had a marker for finding this stuff. But on the other hand, we had to keep getting rid of it, so we had to purify it. Yeah, we think it's perhaps riboflavin. Perhaps.
Your compounds seem to have an amphiphilic nature. Mm-hmm. They have at least 3 hydrophobic tails and a lot of hydrophilic head groups.
Yeah.
So what are, if you just put them in aqueous solution, what kind of structures they are forming, micelles or whatever, and how do they approach membranes? They insert or cover them, anything known?
Okay, let me answer the first question. Okay, so when we did the structure, when we published the structural work in carbohydrate chemistry a couple of years ago, we determined the critical micelle concentration. So because at the time we were interested in biosurfactants, and we thought these compounds would be a biosurfactant. So the answer to that is that the CMC is actually quite high. All right, it has detergent properties for sure. This type I agree with you entirely. It's actually quite lipophilic. It does not extract into chloroform. It does so to extract this. We're extracting to MEK, butanone. And so it has lipophilic properties, but we think it also probably forms micelles. How does that interact with the cells? As yet, we don't know that. We know that the fatty acid chain length, or the number of fatty acids there, seems to determine that in some way. And we know that polyol head group is actually crucial for some type of activity. So we're seeing different activities with different head groups. So we believe there's selective activity. How it interacts, again, we get to this mode of action, which is, again, the question at the moment. Yeah, good question. Thank you.